I want to introduce our speaker, Elsa, if you want to wave to us. Thanks for your patience while we get all our business done for our members. Elsa is a mycologist who's interested in naming and classifying mushroom species in California and beyond.
I just don't know what's causing it.
Oh, could someone mute? So tonight she's presenting mushrooms and their spores. She has described 20 new species in California, or as from California. She works at UC Berkeley and actually has 5 mushroom species named after her. Her main goal is to contribute to the conservation of mushroom species, and for that reason, she's proposed several species for the IUCN Global Database of Endangered Species. I don't know if some of you remember, Todd Osmondson spoke to us about sort of like the endangered species, and we just don't think of that for mushrooms, but it definitely is a thing. She tries to keep current with mushroom literature, and she is an avid knitter, and she likes to use mushroom-dyed yarns for her creations. She said, if I wanted to sum up in one sentence, she's a Dutch cat-loving knitting mycologist. So welcome, Elsa. I had the pleasure of meeting you just north of San Francisco at Soma Camp a few weeks ago. So
yeah, thank you so much for having me from the West Coast. I'm situated in Berkeley, sunny Berkeley, and I am thrilled to see some familiar faces from NAMA and other events. So it's very nice to be here. Tonight, I'm going to talk about mushroom spores, which are fascinating. And, And I'm going to talk about some generalities about them, about lots of exceptions to the general rules, and also a lot about things that we don't know. So if you know someone who would like to study mushrooms, there is so much to learn still about mushrooms. So tell them about spores and everything we don't know about them. So as I said, my talk is in 2 parts, and I'll start with talking about spore essentials and dispersal of spores. And I heavily rely with that on the literature. And I would just point out a series of books written a century ago, I would say, by Buller, who was a British mycologist living in Canada. And he was an amazing researcher. He was such a good observer. It's just amazingly work what he did. And his books are freely available online. And if you have some extra time, just look at them at some point. I'll have the where you can find it at the end of my talk. So his work, of course, is being taken up by other people and more compacted in other books. And recently, there's a lot of attention to spore release, how it goes. The launch of ascomycete spores, and so on and so forth. So there's a lot going on recently as well, and not just only research a century ago. So what you see here are honey mushrooms, and every one of these produces spores. Okay, I should go back. I want to get rid of my stop, and I don't know how to do that. Okay, that doesn't work. Anyway, so here we have a lot of fruit bodies, and every fruit body produces something like 15 or 16 billion spores. I have not counted them. That is more or less what Buller says of a similar shaped, sized agaricus species. And I thought, okay, that will work for these honey mushrooms as well. And you see all this white stuff? Those are the spores. So an enormous amount of spores. And of course, a new individual is formed when 2 spores meet. It's not just that one spore can make a new organism. So spores, you can compare them to seeds, but there are a lot of differences. Spore is much smaller. It's only 1 cell, and it can't form a new individual on itself. But a seed can do that. And a seed is multicellular, can survive longer, etc., etc. So there are lots of differences. But the purpose is the same. They're both offspring. So the individual spores are really very tiny. here we can have them under the microscope. These are not the spores of the honey mushroom, because those are pretty boring. These are a little more exciting. They're colored, they have spines, but they're equally small. So you have to put 10 or 12 side by side to get the width of a hair. So you can't see them with the naked eye, but you can see them when they're en masse, when there are a lot of them. So when everything is right, the spore will germinate, and that puts out this tube, which looks a bit like a root, but again, we're talking about things that are one cell, not a whole thing. So again, this is still all one cell. In the tube, we call a hypha. It will grow. They'll grow and form a little network, which we call a mycelium. But this is still this one spore thing. What we need is a second spore. Here, the one spore, and we need the second spore, or a second other mycelium of the same species, but which is slightly different. It's this whole process that will, that will warrant another talk, but you have to think about something like gender, so it's male and female, but not quite. It's not a 50-50 thing. Basically, it is you can mate with anybody who is a little different than you are. So your chances are much higher than if we're talking about animals or humans. But anyway, we need a second individual spore or mycelium to make a longer-term mycelium that can reproduce and form a mushroom again when everything is right, when all the circumstances are good. So temperature, moisture, and food, of course. There has to be enough food to make these mushrooms, because these mushrooms take a lot of energy, a lot of stuff to make in comparison to this mycelium, which is just these thin threads in the soil or in the substrate. So here we're talking about massive things. And what I'm showing you here is a gilled mushroom. And these spores are formed on the sides of the gills. So here, all these gills, they're full of cells that make spores. Same is true for this polypore thing. So they have tubes, and the inside there are just lined with these cells that make spores. Spines do it the other way. Here's the outside. But again, you make more surface to make more spores, or you make more branches. And again, on the outside, spores are formed, but you have much more surface to make spores than just a smooth one, as this sterium does. But the sterium makes tiny cells really tight, so solves the problem in that way. And then the yellow blobs, again, making a lot of surface area where they can have these cells that produce the spores. Simple as that. You all want more spores in as little effort as possible. So the spores are made on these cells, which we call a basidium, with 4 prongs at the end. So they make 4 spores, and that's it. It's not that they pump out spores all the time. It's just a one-time thing, one set of 4 spores per of this cell that is sticking out on the side of these gills. And we always say they drop their spores. That is not the case. They shoot them off. It's an active, unique process with which these spores are propelled into the space, in this case, between the 2 gills. And this is a time lapse of that process. So here we have a 1-pronged basidium with 1 spore on it. You see the spore is asymmetric, stands on the side, and then it looks like it's blowing a bubble. And this is actually a drop. It's a droplet with sugars in it, and it attracts more liquid. And at some point, this drop is so big that it touches this side of the spore, and that process will give the spore the momentum to be shot off. And that goes like, very quick, and there's no way of seeing that really happening. But there is this little video with, here we have this basidium with 4 of these prongs and 4 spores on it. And this one has to drop. And when I do this in 7 seconds, you see something happening. And there it goes. So it goes like this, and it gets this momentum. And it's like giving a push to a balloon. I mean, you give a balloon a push, it will go a certain distance, and then it will stop. And that's the same with these spores. They go a certain distance, as shown in this diagram. So you cut through the gill edge, the gills. You have here these cells with the spores. They're being shut off. Oh, sorry about that. They shut off, and then they drop down in that space between the gills. And you see immediately that they shouldn't be shot off too fast, too far, because they land on the other side, or hardly, and then they also don't make it down into the space between the gills, because they need to go all the way down and then into the air. So the bigger the spores, the larger the spores, the further they are shot off. If you see a mushroom with the gills really wide apart, it might mean that the spores are really long and they have to be shot off quite a distance, but they can also be short, right? So, but if you have long spores, you need a lot of space for being shot off in that space, in the area between the 2 gills. And this Gomphidius does indeed have pretty long spores for a mushroom. This is 15 to 20 micron, is pretty long. Just like bolete spores, but then long bolete spores. And then also when you look at a polypore like this Fomitopsis, you have to realize that these tubes, which are, as you see here, the pores of the ends of these tubes are sometimes something like ½ centimeter long inside its fruiting body. And so they have to be exactly down. So these polypores are actually quite elaborate engineering projects, I think. So they have to be exactly growing that the tubes are totally down, and otherwise the spores are shot on the other side, or they fall down and they fall on the other side of the tube and don't get anywhere out of this fruiting body. And of course, these fruiting bodies, all what they are wanting to do is getting the spores out into the world. So that is always, just if you see these things which are so common, just take a moment to marvel at them. So, you know, that's what I said. The spores are getting out of the fruiting body and in the wind stream. And then you can even see that. I mean, you can have this drawing by Buller, but you say, well, can I see that? And yes, you can. Let me see by the looks. You can see it happening here. So that's what happens. All these spores are getting out of these fruiting bodies. Okay, beautiful picture. We end with a different group of mushrooms. That are those that form caps and morels that have a totally different way of getting the spores out, a way simpler way, a much simpler way than this shooting off, this unique way that we saw in these basidiomycetes. So these are called ascomycetes because the spores are in a tube, which we call an ascus, and they're shut off, basically. And there's a lid on the top that flies off when the spores and the pressure is high enough to do that, and the spores are ripe. And here we can see that as well. When we blow on such a cup, then you'll see the spores all being shot off and blown away by the wind. So people have done research on how this process really works on a tiny mushroom that's really a few millimeters and has these gigantic SI with colored spores that stick out. So you can really see what's going on. It was really amazing organism to work on. Again, Buller, of course, did that. And then at some point, when the pressure is mounting and mounting, the lid, here, what you see here, that flies off, and these spores are being shot off. And that happens when there's a pressure of something like 3 atmosphere, which people have been telling me that's the same as in a car tire. So in every ascus, you have that kind of pressure, and then it's the lid blows off and the spores are being shot off. And the interesting thing about this is that these spores, these SI bend towards the light. So this little thing on dung shoots them off so that they get onto the grass, and then the cow or whatever can eat it again, can eat the spores, and then the spores go through the intestinal tract of the animal and come out with the dung. I mean, the poop, what you call poop, but then it becomes dung, and then this thing can grow on it again. Oh. And the same with morels, not that they grow on dung, but they shoot also their spores off towards the light. So these SI bend towards the light and then shoot off the spores. Okay, not all ascomycetes look like these flat or surfaces. There are also ones that have these little flasks, so you cut it over lengthwise through. You can see here these SI. And these ones just grow up one by one, shoot the spores off, then shrivel back, and then the next one can grow up into the neck and shoot the spores off. And the same with lobster mushrooms. So here you have this surface, there's all these puckles, these little pimples on it. Every pimple is a ending of one of these fruiting bodies. So again, these SI, one by one, grow up, shoot the spores off, and then shrivel back, and the next one can do it. But of course, this is not the only way to get your spores out into the world. There are a lot of mushrooms that don't shoot actively off their spores in one way or the other. So they depend on other organisms to spread their spores. And one big group of that is the truffles, where the spores are inside. These fruit bodies often have a strong smell, and they're sought after by little rodents, which eat them. And then these little rodents poop, and then in their poop, you find the spores of the mushrooms they just ate. And in fact, when you look at poop of these things, or you look at a fruiting body, often there are a gigantic amount of spores into these things, into the poop of these. And these, when they poop it out, the spores are still viable. So in this particular case, we see still rhizopogon spores, and we see Goetheria spores here. And this is not that animal, but you get the idea. Another way is attracting flies by smelling like carrion and looking like carrion, and then the spores are in the goo of this Azaroe species, this stinkhorn species, and the flies come, and at the end of the day, just this totally naked thing is left behind. Then puffballs, again, is a totally different way of doing it. So the spores are in this ball, and the spores are round and hydrophobic, and they're like dust. And it's really like they need to be pressed out of the fruiting body by raindrops or someone stepping on it or whatever, a branch, and then they go off into the world. So they're not shot off from the basidia as we have seen by the other ones. Those are just dropping off. Splash cups is a different system where the raindrops splash these cups, these lentils, what I always call lentils, where the spores are out of these cups. And there are really beautiful videos on it, but my program didn't want me to insert them into my file, so I apologize. It's only a picture, but please look online for nice pictures of splash cups of these bird nest fungi. Splash cups. So that was my first part, and I wonder whether there are any questions about this first part. I don't know who can look.
Yeah, we did. We had a question about the number of spores per cell when you're talking about the basidia.
The norm is 4 spores, but there are exceptions. There are ones with 2, and there are ones with 6 or 8. Like chanterelles are notoriously high producing. more spores per basidium. And then there are lots of species that have only 2 spores, but 4 is the norm. Agaric, your button mushroom that you buy in the grocery store is an exception with 2 spores per basidium.
Hence the bisporous.
Bisporous, exactly.
And John was asking about, curious about the artist conch and how a lot of the spore deposit is found on the top. And is it defying gravity?
These spores, they have a specific spore wall that is it's a spore wall that is double with little canals in them, and they are very good at ballooning up in the wind, by the wind, and coming down on the thing. Yeah, the Ganoderma spores are crazy, and often also because there's so many and so brown, you often see the fruit body because we see first the spores and then the fruit body. Yeah. And I have a photo of that later.
Oh, great. We had one other question from Benji that said, are there any spores that need to be ingested to be viable? And the word for that he's calling obligatory.
Obligatory.
Thank you.
I don't think that is the case. I think it is more the case that the spores will survive, but not that it is obligatory to go through the intestinal tract of an animal, as far as I know. I know that some spores, and we get to that at the end, their viability improves by staying longer in the soil, which is very interesting. But that is it, yeah. I'm going to put the light on. Okay.
Right. Any other questions before Elsa moves on to part 2? Nope, I don't see any. Thank you.
Okay, we go on. So we can now ask, we know how the spores get out, and are there other adaptations or correlations between other things of the mushroom and their environment? And again, lots of literature about it, about the shape of spores, shape and then the form, the colors, the size, changes in phenology of fruiting of mushrooms. There's a lot going on in this field right now. Okay, spores. So let's look at them. Many of them are just thin-walled, colorless, and really most of them are ellipsoids. That's just the general shape with a length-width ratio of something like 1.3, 1.6 in that area. The length also something between 7 and 10 microns is often the case. Here is an example of, these of course are not boring because it's from my group. They're slightly thick-walled. They have a germ pore. Here they were attached. So these are already quite interesting for my group, but I can imagine that you don't join me in my enthusiasm for Leucoagaricus spores. Entoloma spores are fascinating. They are really like little boxes sometimes. You can see that they are asymmetrical. Like I said, they are actively shut off, so they're asymmetric. This is the side view. This is from the belly or the back. Again, it's a belly back view, side view, side view, side view. And this is just one example, but there are many more other examples of Entoloma spores that it looks like box and maybe more square, or someone put his hand on the top part and someone else and the other hand on the lower part and twist it in different directions, and you get these really strange shapes. So there's a lot going on in that genus. Then we have here symmetrical spores, I would say asymmetrical spores that are symmetrical, so of this truffle species. So they're nuts, but they have these wings and they're also a little bit colored. So that's also not quite boring. And then russular spores, here we have them in water or KOH. So we can see there's something going on, but not really. But when you put them in an iodine solution, you said these spores are really ornamented and stain blue. So it means that there's some sugar or starch-like substance on in the cell wall, the spore wall, sorry. And when you look at, this is a different species, under the scanning electron microscope, you see even more details, and you see networks and all kinds of things. So Russell spores are really amazing too, and Lactaria spores, milk cap spores as well, so really amazing under the microscope. Inospes, not all look like this, but this is a nice example of Inospes with spines, with finger-shaped things sticking out. And these are spores that I looked at, and I can imagine you were not very impressed, but you can see here this germ pore, which looks like someone put a thumb in. Some have some ornamentation, you have some different shapes. So there's a little bit going on there that is interesting to keep myself awake when I look at many of these spores. So there is a lot of difference in the size of these spores. So we can ask them, well, are small fruit bodies always producing small spores and large fruit bodies large spores? And of course, it would make sense, because when you're small, you want still a lot of offspring, and you don't have resources to make gigantic spores. So indeed, yes, in general, you can say that there are large spores made on large fruit bodies and small, tiny spores on small fruit bodies. But it's not a 100 thing, because also it depends on what we were all talking about, from how many spores are produced by Basidium, because there are tiny species that have two-spored Basidia, for instance, Lactaria, and they have large spores. So they put more effort in making the offspring than in making masses of offspring. They're putting their energy into, putting a lot of energy into these bigger spores. And then we can also ask whether, looking at fruit body size from mushroom size, who has the largest spores, and does it depend on how a mushroom gets its nutrients? So there are these two main groups we are probably all familiar with. There are ectomycorrhizal species that, like boletes and amanitas, that give nutrients to the tree in exchange for sugars, for their carbs. And many ectomycorrhizal species do form pretty large fruit bodies, not all. There are also Inospes that make a lot of tiny, relatively small fruit bodies. But yeah, when you look in general, at least in my area, at any table after a foray, the ectomycorrhizal species pile is much bigger than the pile of the saprotrophic living species. So saprotrophic species break down plant material directly to get their carbs. And so it's like mycenas and these little cunipellises, they make a lot of small fruit bodies, but even a bunch of these small fruit bodies don't weigh as much as these big boletes. But of course, there are exceptions. And one of these exceptions is this big thing that Matt Smith found in Florida, a big macrocebae. And this is a saprotrophic species. And this is, again, one of these things that you think, well, how do they do it? How do they do it? How do they make these big fruit bodies while just breaking down material? So I really would love to know more about the natural history of this particular species, and also about agaricuses, for instance. So that's also one of these exceptions. They can form pretty big fruit bodies. So how do they do it? So in general, perhaps we can say that ectomycorrhizal species form the larger fruit bodies, and they also form the larger spores than saprotrophs. But there are other things to think about. Things about the size of the individual. So here we have a fairy ring an old fairy ring with big mushrooms. So this one is really has been there for a while. Again, the question for me is from how can they get so many nutrients that they can make these huge fruit bodies? But this is an old individual. I'm not saying that when it were small that they make smaller fruit bodies, but they probably make fewer. And then on the other hand, we also have species that come up, fruit in huge numbers, and then are gone again. So these early colonizers, which often come in by spores, stay a little bit. And I think that in general, these would make smaller fruit bodies. And this kind of life history, you can have that in saprotrophically living species and also in ectomycorrhizal species. So in the pioneer species that are the first to arrive with a little tree in a floodplain, for instance. So they form often small fruit bodies like Inospes or Lacarias. So there is a lot more going on than just these two strategies of the way you get your food, but also other strategies from where are you in the whole life cycle? What are you doing? And for most species, we have just no idea on how big an individual is, how many fruit bodies it forms. We just don't know that. We know that for perhaps a few things, but not for many. So what do we have? Small individuals with small mushrooms or big individuals with big individual mushrooms? A lot of questions in this field. So we have the mushrooms, they form the spores, escape, get into the air. Where do they end up? Where do they end up? And is there any correlation between where they end up and the spore shape or the spore ornamentation? Spores, you see here already, many don't make it far, but we also know that some can get far away from their place of origin and still germinate. The chances that they meet at that time another of their cohort, another of the same species, are of course extremely slim. We know this because we bait them, but we don't. So those chances are very slim, but they can happen. I mean, you know about long distance dispersal of spores across oceans, like rust spores and dust clouds and things like that. But people have been looking a little bit about at this kind of questions. And the way they have been doing it in my area is this Kabir Pay's lab. They have been looking at mushrooms that grow in pine forests or in little pine islands, what they call them. And these pine islands are surrounded by shrubland, which is made up of shrubs that are not mycorrhizal. So the pines are mycorrhizal, so they have a particular suite of species growing with them. And then they look outside the area where the pines are to find whether the spores land there and are still viable. And they do it in an indirect way because you cannot really look at soil and see, hey, are there spores and from which species are they and how far did they come back? They don't know that. So they did it indirectly by taking soil of a distance, here's the distance away from these pine islands, and then they put a little seedling in it. And what they expect is that very close to the pine, on the edge of that pine island, they find a lot of species on the roots, and then further away, they find very few species. But the question is, of course, on who will get there and who is only really close to the forest edge. So they put the seedlings in, wait for 6 months, and then look at the roots and determine from who they have on their roots. So they got something like 7 species. And we look first at the 2 that are wind dispersed. There's elfin saddle, this Helvella species, and the Cerillus. And really surprising to me is that Helvella is not able to get out of this pine island. They get a lot really close to the edge, but then it drops off and they basically don't find it on the roots. It can be that it is outcompeted by the other ones, but anyway, very close to the to the to the edge of the wood, there is abundant. Cerulus pungens, the cerulus here with the pine, is a totally different story. They get really far away from the edge, something like 1 kilometer. They still find these on the roots of the little pine seedlings. And then the truffles, of course, are being eaten by these little rodents that scurry off into the shrubland and poop, and the spores get into the air, the soil, and the little seedlings pick the spores up, and can germinate because of the spores of the rhizopogons there. So you see something like 10 meters here, something like 100 meters here away from the edge. Those things are there. So not too far, because if you're a little rodent, being out in the open is a dangerous thing there, because there are these red-tailed hawks flying around and other animals that will eat you. And then the last one I will get back to really soon is, I'm mixing this up, is this Tomentella species, and I'll get to that in a few slides. So we have seen already that these ectomycorrhizal species want to end up on their roots of these, in this case, there were seedlings they planted on, but there are saprotrophic species. They want to end up on the substrate. They can break down, and this Gymnopolis, it's eucalyptus debris. This Mycena grows on oak leaves in California as Mycena californiensis. A parasite like this little Octospora grows on specifically on a moss species. They are very particular about which moss species they grow on. And then these ectomycorrhizal species, the spores can only germinate with the tree root when they are close to a tree root, and they give it chemicals. So they sense chemicals of the root being exudated, and then they can germinate. And in this case, this little seedling has already a lot of fungal growth in the soil here. And of course, these spores have been put on this roots and grow there happily. And the same is true for these orchids, these truffle orchards. So here are oak trees in lines, and people have inoculated their trees, and then with hoping that at some point they can harvest the truffles. But there's nobody going out in nature doing that. So the big question is for me, how do these spores manage to get into the soil, to the roots? And the more I walk around, and the more I'm just amazed that these spores actually end up on the roots of trees, because there are leaves, there are needles, there's moss, there's all kinds of things going on. We know we don't know what's going on. It's layered. There are all these different chemical layers in soil. So rainwater is, of course, what you think of first and last and most important to get the spores into the soil. And then the other thing is small animals. And small animals, I think of mites, insects, rainworms, worms, springtails, that kind of things. And it appears that for some we can really show that. And that is for that crust. I was skipping in this picture of these species that get out of the pine islands. And this, you would say, what? Is there a mushroom? Yes, there is. There is a crust growing on the underside of this piece of wood. This piece of wood is lying with the mushroom down on the soil, and this is an ectomycorrhizal species. So it's food for fruiting, it needs wood, but for its nutrients, it's connected to tree roots. And when you look at the spores of this thing, they are knobby. And when you look at the mites that walk around through this fungus, they look like lawn mowers. They just covered in these spores. They eat the spores. They're just spore-bearing mites, basically. And they walk around, and they will also get a little bit into the soil, and they might get to the roots and deposit the spores there. This is a close-up of this Tomentella species we were talking about, and you see all these spiny things on the outside of the mite. So with this example in mind, people have been looking at actomycorrhizal species in general and thinking, well, are there more of these spiny spores? Can we see that there are more ornamented spores in actomycorrhizal species? And actually that is the case, but we shouldn't forget that these Tomentella, there's this crust where these mite walk through it and then walk just into the soil and deposit the spores, basically. That's totally different than having a Cortinarius where the spores are wind dispersed mostly and are hardly, in this case, hardly ornamented. So again, from how do these spores end up where they need to be, and does the ornamentation help? That's a big open question for me. Russulas, the same thing. The spores are ornamented. I mean, the amylate wall, the wall that the sugar coating, quote unquote, I can see the function of that because that will keep the inside of the spore moist. It is like an M&M, the sugar coating on a chocolate thingy. The chocolate stays nice and juicy. And I think that will help. The spore wall here will help against desiccation, drying out. But whether ornamentation helps, that's a totally different question. And in anosopy, we have basically two types of spores, these knobby ones and smooth ones. And I would say that both groups are equally successful in nature. We see both of them. In Amanita, we can see actomycorrhizal and saprotrophic species, and they all have smooth spores. So there's exceptions and there are rules, and there's a lot we don't know why they are. And then when we look at these hydrophobic spiny spores of Pisolitis, I really don't know how these get to where they want to be, because in my area, they fruit in the middle of summer when it is just completely dry. But this is in my backyard. There's no green here. It's just except this one finca that survives, but it's totally dry. There's no insect or any other activity. And these spores do get around, get into the air, and are being deposited somewhere and get to the tree roots because these are actomycorrhizal. So for me, a big puzzle. How does this work? And then again, soil is just a black box, and chemical characters of the spore wall, in combination with those different chemical characteristics of the soil layers, I think they play a much more important role than just the outside ornamentation in itself. And again, on the other side, there are also saprotrophic spores with rough ornamentation. So we have, again, everything. But then there are these things that really puzzle me as well. Okay, keep loading you with my questions here. So there are a lot of wood-inhabiting mushrooms, and the late oyster on the left, and then this, what we call in Dutch, we call this a bacon mushroom, bacon fungus, on the right, and they have sausage-shaped spores. So you only find sausage-shaped spores in wood-inhabiting fungi. Mushrooms. No, I'm not saying that all the ones that grow on wood have sausage-shaped spores, but the other thing around is true. So why? Why do they have these little sausages? Question mark. And then we have these huge spores among tree parasites. These are the radiscus species that grow on the outside of bark. They have gigantic spores. Well, all the other ones have literally tiny, tiny spores. Again, I have no idea why. And then lastly, we can talk about spore color. Does that matter? So many species of gilt mushrooms have indeed white spores, but not all. And this is a complicated figure, and I'm going to get rid of part of this figure. This is a figure that shows you here the tree of all the gilt mushrooms And normally you see these things as just straight. Here it is bent backwards so we can have more on the page. And here's the oldest group. These are wax caps. Go here, we have Amanitas up here. And then on the left, we get Agaricus, we get Cortinarias, we get Inocebi as the youngest kids on the block. And then when you look at the spore color on this figure, you see that we have colored spores here, from here onwards, all the way down, and mostly white spores on the right. With some exceptions, we have the pink spores of the Entalomas and of the Plugiuses. So they are here, but this part is all white spored, and then at some point, there was a shift to brown spores or colored spores, and they stayed with it. And in this group in between, which I still call Agaricaceae, it's just this big group. It's been split up in 4 families, but it's just easier to say, okay, Agaricaceae. I feel there was this experimentation with color. So we have all kinds of colors there, from white, yellow, bright yellow, red, green, blue, brown, and black. And then they stuck with the brown. But here's the green, for instance, Chlorophyllum molybditis with green spores. And here we have dark blue. It's really dark blue, and it's not black, it's dark blue. It's a tropical species from Southeast Asia, and it has a blue spore print, which is amazing. So that happens. But this was just the one thing. And after that, they stuck with boring brown, orange rusty brown in this Cortinarias, more purple brown. And most of these brown things are caused by melanin. And they take up a lot of weight of the spore, these spore balls. Melanin is a perfect protection agency against sunlight, UV light, and drying out and high and low temperatures. So it's very good to have that. So this is in gilt mushrooms. In conchs, we see mostly white spores. Conchs and other crusts on wood, we have white spores mainly. This is the European fomus on beech. Exception, which we talked about already, the Gynoderma, which you saw, you see first is all this brown, and then you see, oh, there are these fruiting bodies. So this is one of those exceptions on the white spore rule for mushrooms, conchs. And it has been shown really that having these colored spores really helps surviving in all kinds of nasty environments. There's no correlation with whether you are a saprotrophic or an ectomycorrhizal species. We saw that in that tree, that phylogenetic tree, that bent tree, and we have, Stropharia and Saturella in the same group as Cortinarias, which is ectomycorrhizal. So we have them, they all have colored spores, independent of what they do for the rest. And my penultimate example is that of those gilt mushrooms on dung. So we have already talked a little bit about these ascomycetes, these things on dung, but it's done a gilt mushroom on dung. And this also has colored spores, really thick-walled colored spores. And they have a germ pore. So they have a weak spot in the spore wall where they can germinate easily, because that's the drawback of having a thick wall, that you still need a spot where your hypha can get out, where you can germinate. And of course, this color, this dark color, helps you survive when you're here in this open area, this open exposed vegetation. And the thick wall also helps you survive in that whole process, going through the 4 stomachs of the cow with all the different enzymes. And it's just a very harsh environment for a small spore like this. So these have these 2 adaptations. One other tidbit of information is a rule that ectomycorrhizal species, their spores, ectomycorrhizal gilt mushrooms, let me say it like that, we don't know of any that has spores with a germ pore. So they don't have that. So perhaps we should say that their spore walls are thinner, but anyway, they don't have germ pores. We find germ pores in saprotrophic living species alone. And then, of course, the last question we really want to know about from how long do these spores remain viable? And then, well, we can say some of these very thin-walled tropical species, we know they remain viable only very short periods, perhaps a few hours. But we also know that some of these rhizopogon spores buried in soil remain viable for a really long time. And a really long time, I mean years, 10, 15 years at least. And these spores are not really looking very thick-walled or anything, but apparently they become even more viable after a few years. And I must say that in this area, with the photo on the background, they wait in the soil for an event that a fire that makes that the cones of this pine tree will open. These trees have pine trees have cones that only open when the temperature is high. So when a forest fire comes through, and then the spores land somewhere in this open landscape where these spores are already waiting, because these spores are everywhere, and they wait for this kind of events, that there are new seedlings coming, new seeds, and they can germinate because of that. So I probably have shown you a lot of information in a short period, but I hope that when you're now walking through the woods and see a russula, you think, oh, those have these really interesting spores with a amyloid coating and an ornamentation, and we wonder why that is. And just think a little bit more about the role of fungi, the role of mushrooms in the life cycle of these organisms. And I also would like to thank everybody who has been listening to me, and those people whose pictures I've been using and slides I've been using, and I've been talking about this subject, my colleague in the Netherlands, for instance. And thank you again for being here. And then my last slide is the literature I was talking about. So this is Buller's book series, Researches on Fungi. And then this article that's also freely available online, Gone with the Wind, is a review of spores of gilt mushrooms. And at the bottom is this little video about splash cups of mushrooms, of these splash cups of the bird nest fungi that splash the spores out. So thank you. And I can stop sharing. Oops, I have to get back to my Zoom.
Yep.
Yeah.
Thank you so much. I have a whole string of questions about spores now. I'm getting back to, I think the first one here was from Cassie. Are there any hard and fast rules for spore compatibility with respect to reproduction? So she said, or could the spores from a single mushroom potentially produce a new mushroom as long as they're a little different from one another?
Yes. But again, that depends on the species. But as in humans, we have two genders that can mate and form new offspring. And fungi, you can have 1,000 or more different. It's not gender, we call it mating types, but you get the idea. And you just have to be different than the other one, basically. But again, that also depends on the species. So no hard rules there either. Sorry.
Fascinating. Cassie, does that answer your question? Heather's asking, is there anything special about the spores of lobster mushrooms or the aborted entoloma that can account for the mutation we see in them?
No, not that I know. No, it's just, it is a parasite that grows over the, so the lobster, what you see, that red thing is the ascomycete, and then underneath is the russula. So there's nothing in the spores of the russula that makes this. It is just, it is a very aggressive parasite.
And Claudette, her comment kind of alludes to that they're have different stories, how they're an asco attacking a basidio and then the basidio attacking another basidio.
Yeah.
So Peter's asking then, do abortiventilloma have spores?
I think someone in your area, because we don't have it in the West here, and we didn't have it back in Europe where I'm from either. So I don't think they have spores anymore, but I hope someone in your area can answer that question.
Yeah, interesting. Actually, Claudette is saying the Guild version does, but the stump dumpling version
does not. Yeah, that makes sense.
Stopping it from releasing spores and reproducing.
Yeah, and the lobster, the thing it grows over, the Russula doesn't make spores. That grows just over the whole fruiting body. Yeah.
And then Peter was asking about when you were showing the truffles and said, are they inoculating trees with spores or
Yes. Yes.
With spores?
Yeah, you can buy little oak plants with the spores on it.
Already inoculated. Interesting.
Yes.
Let's see.
Well, in France, you can buy those. Let's be clear. This was a picture in France.
I had heard that too, that when I think I saw at NAMA a truffle lecture, and he was saying it isn't so much the kind of like the climate here, but the glaciation here that makes it that maybe there weren't as many truffle species here because they didn't have as long to develop. There wasn't, a lot of the stuff was scraped away. That was a theory. Peter's asking, how can we help spores succeed?
By doing as little as possible. Leaving forests to their natural cycles. Let them burn if that is what is normal in your area, and don't introduce any pests.
Someone is asking, when you were talking about spores getting into the dirt, how about animal hooves and claws pushing them into the earth? Or footsteps?
Probably helps, but you have to remember they have to get really into the soil, and a hoof print is not. It's still the surface, right? But I see hoof prints in my backyard from the deer, so I can say that. Yeah.
And then Benji is asking, how similar is the sugar ornamentation that you've discussed to similar post-translational branching sugars and other bacterial and eukaryotic microbes?
Ooh, I have no clue about that. I'm really sorry. That is a question that is beyond my expertise. And I use the term sugar is really kind of in quotes because it is a coating of some, and I hope that someone else can answer this question better than I can. But I have no idea about the structure, exactly what it is and how that compares to other sugar structures in other organisms.
Mike has noted, according to noted mycologist Abraham Lincoln, they can last 4 spores and 7 years. And other people are saying that's some laughs and some that sounds like a dad joke. Anyone else have any other questions? And you can feel free to unmute yourself if any of your questions weren't relayed properly. Anyone? Well, gosh, I feel like I learned a lot. I was fascinated by the tree that showed that the older things were generally white spored and that the colored spores is a more recent mutation. That I find that fascinating.
Yeah,
fascinated me as well. Yes.
Yeah, that was when I discovered that. When I looked at that tree, it was when you put it on it to think, well, whoa, really, that is amazing. So there are the pink spores amid the white spores, but then it's in the Agaricacea, they go brown.
I like this. I liked your summary that they experimented with all these different.
Yeah, that's my.
I like that. It says, okay, brown works.
Yeah, but yeah, there are amazing colors in that group. That it's just when you think, the bright yellow, brown, black, white, red, and then brown.
As a person who does dyeing with mushrooms, do you think the spore color has anything to do with the colors that it produces, or it's other
No, it's other chemicals.
things? Yep.
Yep.
Yeah, because I'm always amazed at the colors that come from mushrooms that you wouldn't think would produce those colors. Some of the ones that are so bright aren't the ones that you get the bright colors from. I mean, some are, but
Yeah. But when hygroscopies don't give any color, for instance, I mean, these nice red caps and things. And then for me, still the amazing one is that you get lilac from an Omphalotus species, from a jack-o'-lantern species, which is just really chemically very interesting.
So I found a few of the Cortinaria smithii.
Yes.
I was with Mayumi, and she said, and you can see on these, like the gills are just, when you hold them in the light, they're just this intense red. Then she said to use the stems separately, that I'll get a better red if I use just the caps, and then I'll get a nice orange from the stems.
That is, I'm not a very meticulous dyer. I don't keep track of what I do. I am it's witchcraft anyway. So just.
I like the throw some stuff in a pot method.
I'm sorry. Science needs other things, but.
Let's see. I see one more question. Okay, Glenn, have there been any biological analysis of spores as to what they are, like what they're made of, primarily proteins or fats or carbohydrates?
Yes, I think they're a mixture of everything a little bit, and I really don't know exactly how these things, but they have to provide the new organism with enough to start. So it's a mixture of everything, is my take on it. And I hope someone like David might be better at answering this kind of questions.
Okay. Gosh. Thank you so much. If there are, let's see, Aaron, his goal for 2023 is to photograph under microscopes as many spores as possible. And your talk has gotten him really excited. I agree.
That is a different world. It's really a new world going open when you do that. Yeah.
Wow. All right. Thank you so much.
Thank you all
so much.
Fascinating.
Thank you so much, Elsa.
Yeah,
That was very, very interesting. Really appreciate your time.